This is a production of Cornell University.
Thank you, Bill, for that very kind introduction. So today I'd like to tell you a little bit about what my lab's been doing for the past number of years. And they've been doing a lot, so I can't cram it all in. So I'm going to tell you about stochasticity and robustness in sepal patterning in Arabidopsis. And I really want to dedicate this talk to my lab. This work is all from their high talent and creativity. And you can see them over the years and how we've grown and changed. So I'm really fascinated by size. I think size is a super interesting characteristic that we often take for granted. If you think about size, it controls function in a lot of ways. So here you've got Wolffia, one of the smallest flowering plants on the tip of a finger, versus a giant sequoia, right? Those different sizes are controlling their function. Or a mouse versus an elephant. I really wouldn't want to meet a mouse the size of an elephant in my house. Organ sizes are really interesting. If you think about the liver in particular, the liver scales to match the body size in humans. And so if you transplant a smaller liver into a larger human, it's going to actually scale up to the body size. Floral organs are super important in terms of their size in attracting pollinators. And cell size, the classic difference in cell size in animals is the different cell size of eggs versus sperm. In plants, there's all kinds of amazing differences in cell size. Here you can just see different size and shape hair cells on this sepal. So size is often worked on by evolution, but we're really in the dark at how this happens. But as someone who loves genetics, this gives me a lot of hope because it means there has to be a genetic underpinning and it has to be fairly manipulable. So if you look at two closely related species, here we've got Arabidopsis thaliana versus Arabidopsis lyrata, and you can see their big difference is in size. They've got the same organs, the same number of organs, but the petals in particular have really scaled up in size. And so hopefully we can actually figure this out. What are the genes involved? Size management is really important in terms of agriculture because size determines yield, but it's not a simple problem. Before I got to Cornell, I was like, ooh, bigger is better. No, it needs to be very precisely managed. And you can see that in terms of this big improvement in yield in the Green Revolution. And you can see the wheat before the Green Revolution versus the wheat after the Green Revolution. The plant is smaller, so you can cram more plants in the same field, and the productive grain part is larger. So we need very precise size management to improve yield. Now, our breeders are fantastic at doing this, but I still think if we understand the underlying mechanisms that are controlling size, we'll be able to make precision manipulations that will be very beneficial. Now, why is size so difficult? It's because it's an emergent property. Emergence is a concept from complexity theory where you think about the whole has properties that are not obvious from the parts. Now, we're in Ithaca and we've had bits of snow this winter, and so we have to talk about snowflakes. So if you think about snowflakes, you've got all these water molecules interacting. They don't have any blueprint. All they know is what's going on with their neighboring water molecule. And that interaction is causing this emergent phenomenon of a beautifully symmetric snowflake. The water molecule over here has no idea what's going on over here, right? It's all due to local interactions that create this beautiful structure. I think this is what we're up against in biology. A lot of the times we've got local interactions at the molecular level, the cell level, and they're producing emergent phenomenon at this cellular and organ and organismal scale that are really important. That's what we've got to figure out, but it's not trivial. So one of the things that I and my lab love to do is watch plants as they're growing and developing. And you can kind of see this emergence process happening. Here we've got a living plant. It's on its side under the confocal microscope. And this was one I actually did back in the day. I imaged it every 6 hours. We've got all of the cells marked with the plasma membrane marker. You can see in gold and their nuclei marked. So you've got one sepal here, one sepal here, one sepal here in this Arabidopsis flower bud. And you can see those cells are all growing and interacting with each other in a very complicated pattern, right? You've got cells that are just growing and the nuclei are getting bigger. You've got cells that are dividing. And yet it's producing sepals that are very regular. So a lot of randomness at the cell scale, regularity at the organ scale. So that comes to the two major questions in my lab and how we're thinking about applications. The first major question is, how are cells of different sizes created during development? And what's intriguing is we found a real role for stochasticity or randomness and probabilistic behaviors here. And to me, that's really exciting. The second is, how are organs made with reproducible size and shape despite all of the cellular variability in size, growth, and division? And so that's a question of robustness. How is something happening reproducibly every time despite having lots of perturbations in the environment in lower scales? And then I want to think about how can we apply this to engineered living materials. Okay, so we're going to start with this first question and look at how are cells of different sizes created during development. And so we've been using Arabidopsis plants and particularly looking at the flower. So here's our flower. The outermost floor organs are the sepals. And sepals have this beautiful pattern where they've got these giant cells interspersed between small cells. And that's one of the things that initially attracted me to sepals, because I'm like, this is just really cool. No one's studying this. What's going on here? The other thing that attracts me to sepals is that there's 4 sepals in each flower. And so you can immediately get at this reproducibility in size, even within a single flower. There's more than 100 sepals in a plant. And so you can really get at statistics of how reproducible are sepal sizes and shapes within a plant. So when we started thinking about giant cells, the very first question we had is, how do you integrate cell division and growth with signaling and how a cell is specifying its identity to get at the pattern? How do these things come together? Because a lot of people have studied signaling and cell identity specification, but not in the context of what's going on with growth. Okay, so very early we found that giant cells form through a specialized cell cycle called endoreduplication. And in this cell cycle, normal cell cycle cells grow, replicate their DNA, grow, and divide. And so they maintain the same DNA content. In the endocycle, cells grow, replicate their DNA, and keep growing and replicating their DNA, so they become enlarged and polyploid. And if we look at DNA contents in this sepal, we can see we've got 2C diploid cells, 4C, 8C, 16, and even some 32C. It's these 16 and 32C very polyploid cells that are our giant cells. And we can see that if we look at cell size versus DNA content, the large cells are these 16C cells. The 8C are in between. We've got the 4 and the 2. Okay, so cell size is scaling with DNA content. So what we'd like to do is track the cells as they're growing and dividing. And we use this image analysis software called morphographics for doing this. And you can see there in that movie, replay it, that we've colored each cell and all of its daughter cells with the same color. And so you can start to see how this pattern of giant cells and small cells emerges. What you can see is that the giant cells started very early and just grew the whole time, whereas the small cells divided into a whole patch of smaller cells. So it's really the timing. Giant cells started endoreduplicating early, so they had that whole time to grow, whereas if the cell is dividing multiple times, it makes a patch of all smaller cells. Okay, so how is this cell size pattern generated? We know it's by a variability in the timing of endoreduplication. We know cells that endoreduplicate early become giant cells because they have this extra time to grow, right? They're just growing and replicating their DNA the whole time. But how does an individual cell make this decision to divide or endoreduplicate? And to get at this, in my postdoc and starting here in the lab, we did a mutant screen for mutants that change the pattern of giant cells. And so we pulled out this whole range of mutants. And you can see we've got things without giant cells, with things with fewer giant cells, and we've got things with more giant cells. So we've got a whole genetic repertoire. Interestingly, we have things in the cell cycle. We have things involved in specification. And I'm really going to focus on this HTML1 gene right here. So what's the purpose of giant cells? People always ask me that. And one of the things they do is control curvature. So when the sepal bud is closed, the sepals are really defensive organs. They're keeping it closed, keeping it tightly closed so that anything in the environment can't get in there. No insects can eat the reproductive organs. So it needs to stay very tightly closed. And then when the flower blooms, the sepals need to straighten up and allow it to open. The giant cells help this happen. So wild type really straightens up. If we have sepals with too many giant cells, they splay outward, and they splay outward too early. If we have sepals with no giant cells, it's very subtle, but they stay curved inward. They don't do this straightening mechanism. And we see a very few defects in flower opening, that they're just not opening quite normally. And the other thing we found that giant cells do is they have a defensive role. They seem to be involved in biosynthesis of glucosinolates. So for those of you who are not in the planty world, think about mustard. So defensive compounds against insect predators. Okay, so my former graduate student, Heather, found that giant cells are specified by levels of ATML1. So ATML1 is a transcription factor. It's involved in specifying giant cells. And in wild type, we have a few scattered giant cells. When she overexpressed ATML1, she gets sepals completely covered in giant cells. If she lowers down the dosage, she can get gradients in between. And then heterozygous mutants have less ATML1 than wild type, so they've got fewer giant cells down to next to no giant cells in the mutant. So it's really, really dosage sensitive. And that tells us that levels of ATML1 are really important in specifying giant cells. So my former postdoc, Vijaya, wanted to address this. And she asked, okay, how do higher concentrations of ATML1 specify giant cell identity, whereas lower concentrations allow that cell to divide? Because ATML1 is specifying giant cell identity and causing the cell to endoreduplicate. And to get at that, we need to know what are the targets of the ATML1 in giant cell pathway. So what Vijaya did was an interesting experiment where she took an inducible line of ATML1 and induced it with different concentrations. So she can get different levels of ATML1 with this line. She can induce it with, well, not induce it, mock, 0.1 micromolar, 1 micromolar, 10 micromolar. So she's getting different concentrations of ATML1, and then she's sampling it over different time points, 8 hours later, 16 hours later, 24 hours later, 32 hours later. So we're finding out what happens at different concentrations of ATML1 over time. And first of all, she just checked that ATML1 is actually induced. We get the best induction at this 24-hour time point, and we also like this 32-hour time point, which is immediately following that when it's still quite well induced. And so then she did RNA-seq to look at what gene expression patterns are changed. And she found the most gene expression pattern changes at this 24-hour time point. So we've got the most upregulated and downregulated genes right there, but we still have an important set that are upregulated and downregulated at 32 hours. So I'm going to really focus in on these two time points. And so what are these genes? So she did some GO-term analysis and came up with things like cuticle development, very long chain fatty acid biosynthesis process, wax biosynthesis, fatty acids. And I'm like, Vijaya, ATML1's in the epidermis, is just turning on up, cuticle biosynthesis. Now, fortunately, she didn't listen to me because I was totally wrong. And she's like, no, these are actually really interesting. And one of the reasons these are interesting is that they have the behavior we would expect for a giant cell gene. They're really turning on only at the high concentration of ATML1. And you can see that here, both in our GO-term analysis, we've got the high concentration of ATML1 here. And you can see at the 24-hour and 32-hour, that's when they're upregulated and they're not at the lower concentration so much. Then she went in and looked at individual genes in the very long chain fatty acid biosynthesis pathway, and again found that they're really upregulated at the high concentration of ATML1 and not at the lower concentrations. That's exactly what we expect for a giant cell gene. So she had this hypothesis. We've got ATML1, it's activating biosynthesis of very long chain fatty acids and is making giant cells. How does she test this? So first of all, she starts by knocking down very long chain fatty acid biosynthesis and seeing if there's an effect on giant cells. So she did a RNAi on one of the very long chain fatty acid biosynthesis genes and found this super exciting phenotype. So here's wild type. We've colored the giant cells according to their cell size. And so you can see the bigger cells are more red and the smaller giant cells are more blue. And you get over here to this KCR1 RNAi where very long chain fatty acid biosynthesis is knocked down. And we're down to just the smallest giant cells. So it really has impacted giant cell development. We've lost the big giant cells. So that was a sign that actually, yes, this is involved in the giant cell pathway. Okay, so we know if we're knocking it out, we're affecting giant cells. So then she did a very interesting experiment where she looked at inhibitors. There's chemical inhibitors of very long-chain fatty acid biosynthesis. And she applied these to ATML1 overexpression. So she's got her ATML1 overexpression sepals, and you can see it's got tons of giant cells in here. And she applied the very long-chain fatty acid biosynthesis inhibitor, cafenstrol. And so here's some of the key giant cells. And watched it. 5 days later, they just all grew. And that's what we expect. But at 7 days, she got something spectacular. You can see those giant cells, they went back and divided into smaller cells. I didn't know this was possible. These cells, are 1632C cells. How are they even segregating their chromosomes to divide? We know they are. These cells are nucleated, and they have smaller nuclei, meaning that they did segregate their chromosomes at least somewhat. Even more cool is this point that we see that not only did they divide, these cells are de-differentiating. They were giant cell fate. Now they're coming back and they're starting to make stomata. So that means they really lost their giant cell identity and they're starting to make a whole new cell type because because very long chain fatty acid biosynthesis was inhibited. To me, that was just spectacular. I'm like, wow, I did not know this was possible, and the fact that they've lost their identity is even more exciting. So we've got HMO1 is increasing the biosynthesis of very long chain fatty acids, which involved in both the adenosine specification and maintenance of giant cell fate, and then also endoreduplication. So what are these very long chain fatty acids doing? We started talking to Chris Fromey about using AlphaFold to look at the ATML1 structure, partly because ATML1 is known to have a start domain. Start domains are domains that bind fatty acids, lipids, fatty acid containing lipids, sterols, things of this chemical nature. And so we're like, ooh, maybe there's some relationship here. So we talked to Chris, and his grad student Saket made an AlphaFold structure of ATML1 for us. ATML1 is a transcription factor, so it has a homeodomain that binds DNA. It's got a zipper loop zipper that's involved in dimerization. It has start domain 1. And then they started talking to us about start domain 2. We're like, what are you talking about start domain 2? There's start domain 1, and then there's this extra, start associated domain. Well, it turns out it has a start domain fold that no one had recognized before. So in fact, there are two start domains. They form this didomain structure. And part of the reason people hadn't recognized this green one as start domain 2 is it has this huge loop in it that kind of folds out. Okay, so it doesn't have just one very long chain fatty acid binding domain, it's got two of them. But how are very long chain fatty acids modulating the function of ATML1? And what Saket did is he started looking at what can he model in the binding pocket. So here's start domain 2, here's a very long chain fatty acid, C18 ceramide containing molecule that he's modeled in the pocket, and you can see it's kind of sticking out. Very long chain fatty acids are very, very hydrophobic. So they don't like water, they don't like cytoplasm or nucleoplasm, and they really would not like this configuration, right? It's sticking out, hanging out there in all the water. Not very good. But then Saket started looking at dimerization, and what he found is that if these two ATML1 start domains dimerize in the start domain 2, it completes both pockets. And now we have the complete pocket for the very long chain fatty acid, for both very long chain fatty acid binding. So it looks like this is involved in dimerization of ATML1, potentially activating it to activate downstream genes. And so it looks like it's regulating ATML1 via this dimerization. So that was a very exciting finding. And if you think about, those of you who are on the planty side, if you think about auxin and how it's perceived, it's a very similar mechanism of, it's the glue between two proteins that leads to degradation. So we're thinking the same kind of mechanism here, just not leading to degradation. Okay, so then we put this all into a computational model, because one of the things we love doing is modeling and seeing how our hypotheses play out in the computer. And so we've got this gene regulatory network based on ATML1, dimerizing when it's binding to very long chain fatty acids and then activating downstream gene expression and undimerizing. This was done by our collaborators Nicholas Russell and Pau Formosa Jordan. And they put this all into their model, and you can see in their model, they've got A2MOL1 fluctuating up and down based on other evidence that I didn't show you today. LEGO's the downstream activator of endoreduplication. And you can see here, you're getting out a pattern of giant cells and small cells. So giant cells are in the purple color, small cells in the orange colors, and you're getting giant cells interspersed with small cells in our model. So this very interesting feedback loop seems to be very important for patterning giant cells and maintaining their identity both. So our overarching model is that A2MOL1 enhances the synthesis of very long chain fatty acids, which then act as signaling molecules to bind to A2MOL1, causing it to dimerize, which activates downstream endoreduplication and giant cell formation and maintenance of identity. Obviously, there are a ton of details I couldn't go into here because I want to move on to question number 2. So how are organs made with reproducible size and shape despite cellular variability in size, growth, and division? We got into this question because we looked at these sepals where we changed the number of giant cells, and it's changing the curvature, but it's not changing the size of these sepals. You can see they're basically all the same size. So what is controlling sepal size? And so if you look at Arabidopsis flowers, you can see that they all have their 4 sepals, and they're very reproducible within a flower, and they're very reproducible across flowers. And so they all have the same size and shape, and you never find something like this, where you've got big and small sepals in the same flower. Same thing, these cute little mice with their ears are all the same size, and you never get a mouse with a giant ear on it. Okay, so this is a really fundamental question, applies to both plants and animals. If we think about agricultural relevance, we've got 2 broccoli heads here. Think about which one you're going to go to the grocery store and buy. How many of you are going to buy the one in red? Okay, no one. How many are going to buy the one in blue? Okay, we've got a fair number of people, and we've got some people who are just not going to buy broccoli no matter what happens. Yeah, most of us are going to buy the one in blue. And that's because it's got reproducible flower bud sizes, right? So this one is not going to be sold, and that's a problem for us on the East Coast, as Thomas Bjorkman has told us about. You tend to get this kind of thing when it's grown with fluctuating temperatures. So there are applications. We've actually been using this for an outreach activity we've been doing with middle school girls, particularly during the pandemic. We couldn't do it in person, so we had to do it online. So we're like, okay, this is a cool opportunity. We can show them the confocal microscope in the lab. So we showed them broccoli and cauliflower. We talked to them about what part of the plant they're eating, which kind of blew their minds. And then we stained it with propydium iodide, which stains cell walls, and put it on the confocal microscope to look at it. And here you can see, here are some of these cells. And so they're seeing how we image things live. And then we did some image processing with them. They could do an online image processing program. So we really had a ton of fun actually showing them something you could never see in middle school, which is what it's like to image on a confocal microscope, and that their vegetables have beautiful cells in them. So how do we get at this process of how organs are reproducible in size and shape? This was done by my former postdoc, Dr. Leland Hong, and we're geneticists at heart, so we took a genetics approach. And we looked for mutants that disrupt this. And we pulled out the variable organ size and shape 1, 2, and 3 mutants, so VOS 1, 2, and 3. These have other names because once we figured out what they were, we found other names. And I want to tell you a smidgen about each one. So Bella in the lab, graduate student in the lab, has been working on the VOS1 FTSH4 mutant. And she's got super beautiful live imaging looking at how these cells grow. So what we're showing you is a heat map here, where we've got the growth colored on the cells based on their growth rate. So if it's growing really fast, it's in yellow, and if it's growing really slow, it's in blue. And these are 3 different samples. And so you can see in wild type, it's got this very nice basipetal gradient of growth. So slow at the tip, coming down really fast here, okay? In her VOS1 or FTSH4 mutant, what you can see is it's really patchy. We've got patches of fast growth and patches of slow growth in the wrong places. And that correlates with how these sepals become very irregular shapes because they're growing too fast here, too slow here. It makes this irregular sepal. So Bella's done a beautiful job showing that. If we look at VOS2, this is the work of my former graduate student Mingyan Zhu and my current graduate student Xuyao Kong. And Xuyao is giving his exit seminar on April 12th. So I'm not going to steal his thunder and tell you what he's done. He's got 2 beautiful preprints on bioRxiv that are in the process of hopefully getting published. But one of the key stories we have here is that timing is super important. Sepals need to initiate at the right time to be robust size. And you can see in wild type, the sepals initiate at the same time, and essentially at the same time, they grow and cover the flower bud, and they keep that same size all the way throughout development. Whereas in the DREAMY1 mutant, which is VOS2, the sepals initiate late, and these late initiating sepals always lag behind and never let that flower completely close. And so it's detrimental to the plant because it's got that sepal lagging behind and it can't close the flower bud. So Xuyao is going to tell you more about that. The one I want to tell you about is VOS3. This is also AS2-7D, and I'm going to use that name from here on out. And this is currently the work of my postdoc, Abhilash. So here's the VOS3 or AS2-7D phenotype. This one's really a shape variability as opposed to a size variability phenotype. We think it's kind of spectacular. So you can see in wild type, you have very smooth sepals. And so we're looking down on the inflorescence where you've got the meristem in the center and you've got the young developing flowers here. And as you move out, you've got the older ones. And you can see here, all of these sepals have lots of folds and wrinkles and outgrowths on them. So they're not smooth. If we take a closer look at that, here's wild type. You can see it's very smooth. Here's AS2-7D. You can see these folds, and you can also see outgrowths on it. And so the key question is, how are these lumps formed in this AS2-7D sepals? Wild type, if you look at a cross section of the sepal, we've got our outer epidermis, we've got our inner epidermis, we've got our mesophyll in between, and it's very smooth. In AS2-7D, the outer epidermis has all these wrinkles, the inner one's normal, and you've got the mesophyll in between. So we thought about buckling, mechanical buckling. Buckling is bending or giving way under pressure or strain, and that's how you might get curved surface, right? So here's a train track that buckled due to an earthquake in New Zealand. So Abhilash live imaged these to see what's going on. And so you've got wild type, you can see it starts off smooth. AS2-7D starts off smooth. Here, wild type's still smooth. AS2-7 still smooth in day 2. Here you can see wild type still smooth, and AS2 has got 2-7 has this lump right here, and you can start seeing the creases on each side. You see it gets more complex. We've got these indentations, and we've got these outgrowths forming. So very, very complicated shape, okay? So if we think about mechanical buckling, what does it take to do for a tissue to mechanically buckle? We worked with our longtime collaborator, Arezki Budud, and his associate, Anna Maria Kiss, on modeling this. And we're modeling this in a cross section of the sepal. So we're trying to model like this inner epidermis, this outer epidermis, and the mesophyll. So inner epidermis, outer epidermis, mesophyll in between. Okay, and so we're looking over time. And we've got in this model that the outer epidermis is softer than the inner epidermis, and also that it's under compression, meaning it's trying to grow more than the rest of it. So it's buckling because it's trying to grow too much and it's starting to wave. Okay. And so we needed to see if that was what was going on. So for the first time ever, Abhilash imaged so that we could see the inner layer of the sepal, because we need to compare the growth on the inner layer and the growth on the outer layer. We're expecting the growth on the outer layer to be more, causing compression. Okay, so we can segment the cells on the outside. Here he's going in and deleting out the inside of the sepal, and we're getting our first glimpse of what that inner epidermal layer of the sepal looks like. You can see this is painstaking work that Abhilash has done, where he gets in there and very carefully deletes out the inner part of the flower. Okay, so now he's deleted more. Uh-oh. There it goes. It's not playing quite right. There it goes. Okay, so now he's deleted all of it out, and then he can detect all these cells on the inner side and the outer side. So here he goes, detecting all those cells on the inner side and on the outer side at the same time. So we can then look at cells over time and measure their growth rates. So we can know what's going on in the growth in both the inside and the outside, which is spectacular. We've literally never been able to see this before. So if we look at heat maps, and here we've got fast growth in red and slow growth in blue. In wild type, what we see on the outside is we start with fast growth at the tip. Later it moves down, as you saw in Bella's results, but it starts out at the tip. Here we can see on the inside, the whole thing is growing really fast. This isn't quite what I was expecting. I was expecting these to grow at the same rate so they'd be flat. What happens is that sepal starts out very, very curved, and it needs to grow faster on the inside to flatten out a bit. If we look at our AS2-7D mutant, it's kind of got more even growth pattern everywhere in the outer surface and also on the inner surface. It is still faster on the inner surface in this light. So it wasn't quite what we were expecting based on our model, but hold on. Then we started thinking about the growth direction. Which direction are these cells growing? So if we start with an initial cell and it grows equally in all directions, it's isotropic. And so we put a little line in it, but it's a little line because it indicates that it's the same direction. If we have anisotropic growth, then it's growing more in one direction than another, and then it's going to get a long line in this direction. Okay? So we look at that in our images. And you can see we've got wild type. Here, we've got all these little lines for the growth direction in yellow. These cells are growing up in the outside and on the inside. So both are very, very coordinated. The cells are growing upward. If we look at AS2-7D, here's what's spectacular. The outer cells are not growing up. They're kind of messy, but they're growing much more in width. But the inside cells are still growing up. So we've got this conflict. The outside is trying to grow too wide while the inside is constrained. And that's how we're getting this compression. It is overgrowth, but it's overgrowth in width. It's growing too wide. The inside is constrained. That's going to cause the buckling. So it fits with our model. It looks like the outer epidermis is under compression due to its growth relative to the rest of the sepal. We've also tested that the outer epidermis is softer than the inner epidermis and shown that that criterion also holds. So to summary, you need coordination of growth across the organ from the outer to the inner side to get smooth organ growth and smooth morphogenesis. In wild type, that's what's happening. In the AS2 mutant, we've got too much growth on this outside, particularly in width, and that's causing buckling and these spectacular shapes. Okay, so let's get on to the applications a little bit. We're interested in engineered living materials. So I am part of the Engineered Living Materials Institute at Cornell. Here's some of us. And this institute, we're interested in thinking about how can you take the amazing properties of living organisms and use them as materials in our engineering. If we think about what we're doing now, the way we live is not sustainable at all. Obviously, plastics are an epic disaster. But even the buildings up here that look so nice in the skyline, they are also environmentally not sustainable. If we think about sources of greenhouse gas, manufacturing is a major part of it, 31%. And if we think about what is contributing to that, iron and steel and cement, which are our main building materials, are a major part of that. Okay, so we need to really think about how we make materials. I would say we must completely reimagine all of our materials and their manufacturing going forward. So we've got a lot of work to do. This is not going to happen overnight. This is our grand challenge for the future. How are we going to make life sustainable? And so for the Engineered Living Materials Institute, we're interested in thinking about how you use living materials to do this. And part of the reason is living materials tend to be biodegradable. They have amazing properties. We were talking about their emergent properties. So how can we use that emergence? They tend to self-repair, right? If something's injured, it often can repair. So self-healing. And so we're thinking about ways in which we can do this. Can we use them in wearables? Can you be walking down the street with some living diatoms in your shirt? Thinking about our habitats, our built habitats, like our indoor air spaces. Here we're thinking about a lava tube, so we're thinking totally futuristic, but with all these organisms and a coating on it that are purifying the air and making it good for people to breathe. Can we grow our own houses? Obviously not at this point, but this is the grand vision of what we might get to. And we're bringing together people. This is what I love to do, as you can tell, is bring together people across disciplines to try to use our brains together to come up with solutions we can't get on our own. And so we've got obviously people from the School of Integrated Plant Science. We've got people from architecture, government, biomedical engineering, mechanical and aerospace engineering, lots of engineering, bringing us all together to try to tackle this grand challenge. So one of the things we're doing, starting new, is working on diatoms. And this was really brought to the lab by Trevor Cross, a new postdoc in the lab. He's supported by the Engineered Living Materials Institute. And so this is in collaboration with Meredith Silverstein and other members of the Institute. And he's interested in these diatoms that chain. So this is Skeletonema grethe. And these are some images that Trevor took that are absolutely spectacular. You can see what's interesting about this diatom is that it makes these silicate-based frustules. That's a characteristic of diatoms. They make silicate-based frustules. And so that's immediately interesting from a material standpoint. You've got this cell wall with silica in it. Here he stained the cell walls, these silica-based cell walls, with a stain, a fluorescent stain, and we're also looking at the chloroplasts in red, okay? You can see in these cell walls, these cells chain together by making these intercalary processes that connect one cell to the next. And they're kind of holding hands at these junctions here. That raises a ton of basic fundamental cell biology questions about how do these cells have this morphogenesis? How do they make these structures? Traditionally, these silica cell walls are made inside the cell and exocytosed. Are these processes made inside the cell and exocytosed? Maybe not, because we see dividing cell like this, where it's made its frustule caps, but it doesn't have the extracellular processes yet. But that's one of the big questions we've got to figure out. The next question is, what do we do with these? What can we use them for? They look like little tiny devices, which is why we think they're exciting. Can we use them living? Can we use them dead? Can we use them to feed other organisms that we want to grow and do things? And so I would love your ideas about what we can use these for, because this is the big creative part of the Engineered Living Materials Institute. How do we use these? But yeah, right now we're starting building a synthetic biology toolkit with a Cal's Moonshot Seed Grant for these guys. Another project that's funded by the NSF, it's a Rules of Life Emergent Network Grant together with Jenny Sabin. Jenny Sabin's in architecture, and she's founded the new Department of Design Technology here at Cornell. And with her student, we're trying to think about how we can use our images to inspire design at the architectural level. Now, if you're thinking architecture as blueprints and housing construction, you're out of date. That's not what we're talking about. We're talking about really innovative structure creation. So Jenny's doing this with her graduate student, Ivania, and in my lab, Byron's contributing to this. So we're thinking about taking images of actually the floral meristem. So here's a beautiful Arabidopsis floral meristem, and how can we use that to inspire design? So here's one of those floral meristems imaged over time. It's every 6 hours, and you can go from a floral meristem that has no primordia on it. You can see it's growing, and it starts making a sepal right here. So we're looking at the emergence of that sepal. Okay. So we take the data, we analyze it in our morphographics image analysis pipeline. That gives us a computerized mesh, or like the structure of the cells, and we can segment the cells and detect each cell. It also gives us a whole ton of data about where these cells are and what they're doing. Then we put that into the architectural software called Rhino and Grasshopper. And then they can model our data in their software. And architects are really good about thinking about structure. What is the mechanical properties of these structures? What's going on here? And then they take that and they abstract it and think about how can you then apply it, say printing plastic on fabric to create interesting structures. So Ivania took our data and we were looking at our wild type flower versus our DREAMY1 mutant. DREAMY1 is one of our robustness mutants. It's got problems in initiating sepal primordia. And so she just started putting this in her analysis pipeline and started thinking about the middles of cells and how they move as the tissue grows. And so she looked at wild type over here, and she noticed that when the cells divide, you've got a whole line of cells that are dividing in the same orientation. It makes a little zipper. Then she looked at DREAMY1, and she saw that the cells were not making this zipper. They were dividing in all kinds of different orientations. How many times have we looked at these images? We never saw that. So it was Ivania with her intuition for how things are expanding that caught something that's different between our wild type and our mutant. So how cool is that? So then she's taking that and what she's doing is she's stretching material. So she's stretching material, making it stress, and then using printing to print on that material. And she can print with different plastics with different stiffness. So she can print with a plastic that's pretty soft in black and plastic that's pretty hard in red onto that stretch material, release it, and see what kind of structures come out. So here you can see, here's the meristem that she's working with, and we're thinking of these cells as being, in red as being the stiffer cells, and then the rapidly growing cells in the middle of the meristem, in the middle of the sepal, are what are, in softer plastic. And so she prints that here, and you can see, so we've got the stiffer cells around the edge, we've got the primordium here, and she's printing it in small bits. And I didn't understand until recently why she's printing it in small bits. It's because her printer won't print bigger sections yet. That's the next challenge. But so she's printing out these bits of cells that she's abstracted, right? She's made them into much simpler structures and seeing what comes out of them. And now, how is this architecture? Well, think about scaling this up to human size. And here she's done that. She's thinking about what would it be like to have this thing form a structure, like a kind of tent that you're walking through with these really interesting folds. And so you can see this as a very innovative, artistic space that's beautiful that you might walk through. This is our next challenge. Ivania is starting to try to think about scaling up to table size. And we'll see what happens with this. It's going to cause all kinds of interesting questions. Okay, so to summarize my talk, our grand challenge is understanding emergence in biology. How is it that small local interactions create patterns at bigger scale? Very long chain fatty acid biosynthesis is required for maintenance of giant cell identity. That was quite a surprise and really interesting finding. Coordination of growth across the inner and outer axis is crucial for flat sepal morphogenesis. How do you get those flat sepals? And we're working on using plants and diatoms as novel engineered materials, living materials, and we're thinking about bio-inspired design. So we're taking a broad concept to this. How can we apply this in a very different context that's new and exciting? So I want to thank the people who did the work. I've mostly mentioned them, but Avalash did the work on the AS2-7D. Leland did the original mutant screen. Vijaya found the very long chain fatty acids. Trevor's working on diatoms. Byron's working with the architects. And Mingyuan did the original work on DREAMY1, and Shu Yao is doing the continuing work. Obviously, we love to collaborate. We've had a whole ton of fantastic collaborations to make this happen, particularly with people who know things that we don't, very different fields. So Richard Smith is a computer scientist. Arezgi and Anna Maria Kiss did the modeling. We've been working with people who know proteins and lipids, and it's really been a ton of fun. Now, I've been super fortunate to have funding from the NIH for the Robustness Project and from the NSF for the Giant Cell Project and for this crazy work we're doing trying to bring biology and architecture together. So thanks, and I'd be happy to take any questions.
So for the very long chain fatty acids, does the composition of those matter for HTML? Like, could you make like a screwed up very long chain fatty acid or put in like elements that are not supposed to be there and screw up stuff?
So that's a very interesting question. What kind of very long chain fatty acids will actually bind to HTML1 and which ones won't? Unfortunately, we do not know the answer to that. Our experiments trying to test that have not worked. HTML1 is actually a very tricky protein to work with. What we do know from Ola's work and Catherine Strick's work is the close paralog PDF2 seems to bind to lyso-PC 18:1 and also ceramide, C24 ceramide, and some other things. And so it seems like there's probably a range, but I would bet there's some specificity too. And we just haven't been able to biochemically figure that out because it's actually really tricky. But yeah, I would totally love to know that. Yeah.
Thanks for a great talk. I was wondering what kind of an environment diatom would need to live.
So they can live in pretty much any aqueous environment as long as they have light. So that is one of these issues with these engineered living materials. We're going to have to provide water somehow. And so that's one of the things we're going to have to really think about. Water and energy, as Laura Gunn always says, we have to worry about the energy. So yeah, we're going to have to think about how do we keep them wet while having the properties we want, which is usually dry. We got a lot of cool problems to work out.
I mean, I missed out on the modeling aspect of ATML1. So it makes sense that as the concentration increases, they can dimerize with and regulate fatty acid biosynthesis. But what happens when it's monomers? What does it do?
So in our model, the monomers are not active at all. And we assume that there's a basal rate of very long chain fatty acid biosynthesis anyway, because of course there is in the cell. And so we really conceive that there's some level of dimers even without HTML1 upregulating anything. And then it launches this feedback loop where it can upregulate. But in our model, it's only the dimers that are active.
Going along with that question, is that only the start2 domain? Like what is the start1 domain doing? Does that also bind fatty acids?
Yeah, so that's in our structure modeling, in the configuration that comes out of AlphaFold, the pocket's kind of occluded. And so we actually can't, it doesn't look like it's binding in that structure. But the question is there a conformational change where it might actually be binding? And we don't know the answer to that. I think there's a whole ton of research to be done on these, looking at what do they bind? Do they both bind? Do they bind different ligands? Do they bind the same ligand? In what context do they bind? So I think there's a ton of cool stuff to probe there. Yeah.
Any other question? Yeah, I have a question.
Oh.
Or somebody else?
No, online.
Oh, online.
I think it's Jeremy.
Okay, Jeremy asks, regarding the HTML1 project, what experimental evidence is there for the lipid-mediated dimerization? Does the C18 ceramide get extracted from the membrane or synthesized locally? We do not have direct evidence for the lipid-mediated dimerization. We do have evidence in vivo of dimerization. We've got co-IP showing dimerization, but we have not been able to get that it's lipid dependent. That's a piece that we haven't been able to get. We do see dimerization. Okay, so that's the whole protein. We see in yeast, if we do yeast-2 hybrid, we can get dimerization of the start 1,2 didomain with the whole protein. So it does suggest that the start domains actually will dimerize with the protein because also we have the zipper loop zipper domain, which is also a dimerization domain. And probably both of those are actually dimerizing the protein, but it looks like the start is important. Okay, and the second part of the question is, does the C18 ceramide get extracted from the membrane or synthesized locally? That is a big question. And I think that's why our biochemistry is failing. What we're trying to do is just mix our purified start1, start2 domain with liposomes and hope that something happens. And we get evidence that maybe something little is happening, but it's not really, it's not reproducible or consistent. And as soon as we up the concentration, we are probably getting some aggregation of liposomes. I think there's probably lipid transfer proteins that are involved. The JS sees that when she induces with HTML1, there's also upregulation of lipid transfer proteins. And I think that's what we're missing. And that's what we're going to need to get this to work is my gut feeling. But that's a whole nother project. So yeah, so cool things to look at. Eric.
Oh, last question.
Yeah, go ahead.
Somebody else.
Yeah, you go for it, Marie.
Oh, sorry, Eric. Is HTML1 in the nucleus and is it exclusively there? Or do you have any other subcellular localization?
We only see the fluorescent protein in the nucleus, which then raises the question of where is it coming in contact with the fatty acid and how's it extracting it from the membrane. A couple different possibilities have been floated. One is that it's coming from the nuclear envelope. The second is that when it's biosynthesized, so Aman Husbands has also seen these two start domains in HD-ZIP class 3, and his hypothesis is that it's actually getting picking up the lipid out when it's getting biosynthesis near the ER. I don't think that's the case here because I think it's much more dynamic. So my guess is these lipid transfer proteins are bringing it from somewhere, but whether it's the nuclear envelope, I don't know. Yeah, so all kinds of cool questions.
Yeah, then last question over there.
I was going to ask about what was known about start domains in dimerization in other transcription factors, like the one that Catherine works on. Is that lipid-mediated dimerization, or does it work differently?
So, okay, so first of all, in animals, there are no start domains in transcription factors at all. This is a plant-specific thing. They're found in HD-ZIP class 3 and class 4. ATML1 is a class 4. Saket did some really interesting modeling of this one, and he finds that only things closely related to ATML1 appear to have this dimerization around that start domain. So ATML1, PDF2, and some of the very close paralogs do it, but even things further out like GL2, which is a little further out on the family, that also looks like it dimerizes with start domains, but in a totally different configuration. And then the further out don't seem to dimerize with the start domains at all. If he looks at the HD-ZIP class 3, they don't seem to dimerize with the start domains at all. So it seems like it's very restricted to just these closely related proteins with ATML1, which is super, super fascinating. It shows that we have to think about different HD-ZIPs doing different things.
Okay, since Adrienne is going to teach us, thank you so much.
This has been a production of Cornell University, on the web at cornell.edu.